upon a time, there were two evil scientists who hated each other.
Hello, I am evil scientist Tom. I hate evil scientist Guzenko with a burning passion.
Hello, I'm evil scientist Guzenko. I hate evil scientist Tom.
Now you see, they were very evil, so they were constantly trying to devise new ways to take over the world. Today, they're both trying to create diploid cells of mass destruction. I'm Secret Double Agent Spy Emma. They both think I work for them. My true goal is to have them both create the two most evil gametes in the world, a sperm cell and an egg cell, and combine them to form a zygote of the most evil organism ever. Hello, evil scientist Tom.
Let me. No, where did I go wrong? I can't make this diploid cell. Help me, evil scientist, spy person.
Have you tried making a haploid cell?
What?
A haploid cell. It has 23 chromosomes instead of 46, so you fuse it with another cell to create a diploid.
Brilliant! I will make this haploid cell that you speak of by meiotically dividing one of my cells into 4 new cells with the processes of Meiosis 1 and Meiosis 2. And then I will become the most evil commander person in the world.
The scientist Tom is already creating the first of my 2 haploid cells. Now to Guzenko. Oh, evil scientist Guzenko, have you heard that evil scientist Tom is creating one of the most evil haploid cells to ever walk the face of the earth?
What? No, I cannot be outdone. I must make one too. But how?
Through meiosis, of course. The 2 stages, meiosis 1 and meiosis 2. First, you take a cell that's already undergone interphase, and then you begin the cell division with prophase 1.
Prophase 1? Aha! When DNA coils into chromosomes and spindle fibers appear, and the nuclear membrane disassembles?
Yes, sir, the very same. And then you take these 2 chromosomes in homologous pairs called tetrads and line them up next to each other through the process of synapsis. Then portions of these chromosomes break off through the process of crossing over,
which allows for the exchange of genetic information, or genetic recombination, making me have the most evil and destructive cell in the history of the universe?
Get to work! That's evil scientist Tom. Hello, Tom!
Oh, double secret agent Emma. I've just prophase 1 my cell, and now I'm on to metaphase 1, where I shall line up the tetrads randomly across the midline of the cell, and then spindle fibers will connect to the centromeres at the ends and attach themselves to the poles.
Right. And then you'll move on to anaphase 1, where the spindle fibers will move the chromosomes to the opposite sides of the cell.
Which will result in genetic variation and independent assortment and me becoming the most evil scientist ever.
That's right. Back to evil scientist Guzenko. How's it going, oh Master Evil Scientist?
Great. I've moved on to telophase 1. The chromosomes are moving to opposite sides of the cell. Look, the cytoplasm is about to divide through cytokinesis. Each new cell will have 1 chromosome from each homologous pair.
He's almost done it. He's almost done the first stage of cell division. Now on to meiosis 2. Now remember, you do not copy the DNA before meiosis 2 like you did before meiosis 1. You start off with the first stage, prophase 2. Now form those spindle fibers and start moving those chromosomes.
I'm on it.
Oh, good to see you, secret double agent Emma. What are you doing here? Why do you keep on moving around?
I don't know.
Oh, well, never mind that. I've metaphase 2, and I've moved the chromosomes to the midline of my 2 new formed cells. See? Wonderful, right? Then I anaphase 2 it. Separated the chromatids and moved them to their respective poles.
You're almost there. All you have to do is telophase 2 and cytokinesis 2. You form a nuclear membrane around the chromosomes, and then you split it in half again. Now you have 4 cells because you're in the second stage of meiosis. They're haploids, and they each have half the number of chromosomes as the original.
I've done it. Now I shall be the ruler of the universe.
No. Now I shall take these 2 cells, 1 egg and 1 sperm, and combine them through the process of sexual reproduction to take over the world!
Oh no, I created my haploid cell as though it were made in the testes. And And as it was a sperm cell, or spermatozoa, I meiotically created 4 haploid spermatids through the process of spermatogenesis and hoped that they would grow into the most evil sperm cells ever.
Oh, yes. And evil scientist Guzenko over here, he created his cell through the process of oogenesis, creating a mature egg cell, or ova, by dividing the cytoplasm unevenly during cytokinesis 1 and cytokinesis 2. Now I have an egg cell and a sperm cell, and I will combine them through the process of sexual reproduction.
No!
Yes.
By the way, sexual reproduction is the production of offspring through the union of a sperm and egg. Secret double agent Emma is now producing an organism that is genetically different from its parents because of the different ways genes are combined throughout meiosis.
This allows for organisms with advantageous genetic variations to survive, and those without them to die. No!
Yes. Ha! I created it! The most evil organism ever.